Let's start off with the second session of this morning. We're going to have some exciting stories. By the way, my name is Marcel Nele. I'm heading the genomics pillar of the Cross-omics project. And 2 years ago, I was appointed as the head of the genome diagnostics in Utrecht. So in that sense, it makes sense to lead the genomics pillar. My co-chair.
That's Martin Gira from the Leiden University Medical Center. Hello.
And let me introduce the first speaker. A long and a good friend of mine. We had a lot of gin tonics over the past few years at the AGBT meeting, which is a typical meeting for tech buffs like Alex is. So he pioneered in a lot of technologies that we had to introduce into the genomics pillar the past 10, 15 years or so. And the latest is long-read technologies in sequencing, but also in visualizing long-read imaging equipment. So, Alex, the floor is yours, and I look forward to your talk.
Thanks so much, Marcel. Thanks all for joining, also for joining online. And indeed, I'll try to share some of our excitement about some of the latest technologies, mainly the long-read technologies. And as you mentioned it, now I can disclose, I always had straight tonics, and you had gin tonics. At least that's the footnote formally for our employers. So if I may sound excited about some of the stuff that I try to show you, I hope you believe me, it's not only because some of the companies shared reagent costs with us for some of the experiment, not even all that I'm showing. What I'm really showing today are two technologies. One is called optical genome mapping, provided by BioNano, or formerly known as BioNano Genomics in the US. And then I'll show some of the long-read genome sequencing data provided by PacBio's technology. And I'll end with a small vignette about why we actually do this, especially in the rare disease space. So for those that have not actually followed the DNA and the genomics field, there is a proper long-read revolution ongoing, yet another next-generation sequencing revolution. And if you just want the single-page summary of that, a little bit of the history, I recommend this milestone paper in Nature from 2 years ago. Also, our very own facility, previously with Marcel, now with Janneke and Ronnie, is pushing long-read technologies. And I think both Janneke and Ronnie are around if you have more detailed questions, if you want to join forces with us and our facility. We've invested quite heavily in long-read technologies over the last years. So there's all details given on this slide. So some that are working not necessarily on genomics may want to take a screenshot of this slide once it's fully animated. It's really these two technologies that I tried to show today. So one is optical genome mapping. And just as a full disclosure, this has nothing to do with sequencing. While we all embrace in the genomics field sequencing DNA, there's other ways of literally looking at DNA, and that is what optical genome mapping offers. Some, including myself, call this also next-generation cytogenetics. As equivalent to next-gen sequencing that replaced a lot of the Sanger test, I predict that this will replace almost everything we do in cytogenetics. That means we look extremely long DNA molecules. The longest ones we see are 2 megabases, so 2 million base pairs in size. We can perform a proper assembly of genomes, and in our case, even of human genomes, and then check in that assembly whether there's any structural variants. So events that are larger than 300 to 500 base pairs, where the structure changes by deletion, insertion, inversion, translocations, and so on. And I think that's a very powerful tool, and it really gives us access to regions of the genome that previous technologies never allowed. In parallel, and the second half of my talk will be about the real sequencing, long-read sequencing. All long-read technologies have something in common, or a lot of the things that are on the slide, they have in common. It's always a single molecule approach. You're literally sequencing just one molecule at a time, different from Illumina and other short-read technologies where it's the average of 10 to 20,000 molecules. And quite astonishing, the technology is now mature enough, you get accuracy, especially with hi-fi sequencing provided by PacBio, that is on par, or in parts of the genome, even outperforming Illumina's quality. But it's Illumina quality, but the reads itself are 20,000 base pairs in size, opposite to 200 base pairs or so with Illumina. So it's a 100-fold enrichment or increase in read length. And that's truly amazing. Because you sequence native DNA molecules, you also get some freebies on top. For example, methylation can be read out by the data that we get. And the length is long enough to do at least partial de novo assemblies of full genomes. So that is important for human genomes, but if you work on any other species, metagenomics, that's really a proper game changer as well. And similar to the optical genome mapping, the long-read sequencing for humans has much, much higher sensitivity, again, to structural aberrations. And I think we, as a field, have underestimated the wealth and the complexity of that over the last decades. And we now reach into regions where Illumina and other short-read technologies never reached into. So now I'll follow with brief pitches about what these technologies are really about and what they can do for us. Starting with OGM, those are really looking at ultra-long DNA molecules, similar to what cytogenetics and karyotyping always did. Many, many years ago, I was even trained in a cytogenetics laboratory, but I never mastered the art of looking at chromosomes and seeing whether a single band in a chromosome was missing or too much. But with OGM, even I can do that. We now take images, and instead of having chromosomal bands labeled, we have a 6-mer DNA motif that is labeled. We have very high precision in how often this motif is labeled, and that's literally what you see. Each blue line is a single, very long stretch of DNA labeled with a 6-mer motif that has an average distance of 5 kb across the human genome. And the beauty is, instead of having a single band on a chromosome by karyotyping, we now have thousands of bands equivalent, but we can digitalize those, count those, measure the distance much more accurately. And that, in essence, is what optical genome mapping is doing. So where karyotyping has usually 500 band resolution, we now have 500,000 bands, but we can also measure the distance much more accurately. So that's why I call it next generation cytogenetics, because it still is similar to karyotyping. A genome-wide analysis gives us positional information, and in essence, we're still looking at single pieces of chromosomes. So a few years ago, when we saw the technology popping up, we were amongst the first in the world using this, and we did proof of concept studies, retrospective studies, to provide evidence that this can be as good or even better than standard of care cytogenetic approaches. So that is karyotyping, CNV microarrays, and FISH. And we reached 100% concordance for somatic structural variants in cancer, but also for germline aberrations in constitutional aberrations. This was so promising that our management funded another prospective study specifically for one subtype of leukemia, that is AML, and there it clearly outperforms in all aspects what standard of care could do for us. So we think AML is a specific field where this should enter the diagnostics pretty soon. So that's more diagnostic view. Now looking at research, we're working a lot on rare diseases where we know that mutations often happen new in each generation, so very severe diseases that are sporadic. And what we then do for a lot of our technologies is approaching patient-parent trios. And the reason is that we know that for severe rare diseases, de novo mutations that happen only in that one oocyte or sperm cell, in that one new generation, those are the important mutations. And if we then perform the genomic data set, not only on the child, the patient, but also the unaffected parents, we can deduce the actual newly occurring mutations quite easily. If you look at de novo mutations from a more technical standpoint, they are the most challenging mutations that you can work with, simply because a de novo mutation in the child looks exactly as all the false positives in the child. And usually the false positives are much, much more than the true de novo mutations. So if your technology is not up to speed and not very accurate, you can't do that very efficiently. And here we've used this trio approach in particular for BioNano. There's also a lot of predictions that de novo structural variants in particular are the key to the regulatory part of the human genome, and that's clearly what I signed up for. I think that will be the discoveries of the next 10 years or so. Just to highlight how BioNano data look like, this is a so-called circus plot where you find the roughly 7,000 structure variants you see in an individual genome. See how many of those are insertions, deletions, some inversions, duplications, and translocations. So that's a single case experiment. If you use an inbuilt database of 300 controls, we can reduce that to 30 to 50 events per genome. But even if you're right that one of the structure variants is causing a disease, you still have to figure out which of the 30 to 50 it is. But if you have the parental data on top, you can just reduce out everything that is inherited, and usually you're left with 0, and occasionally we're left with 1, as in this example. So from this, we already learned 2 things. The de novo mutation rate for structure variants is exactly very, very low. There's only very few of those. And secondly, this technology is very accurate, otherwise we couldn't do this effort. And I just brought one specific case from this filtering strategy here. You may wonder what kind of structure variant has been missed. These cases underwent all the other genomic tests in the past. It's actually shocking to learn that there's a very large deletion. It's 121 kb in size, so 120,000 nucleotides in size. A lot of the previously used tests should have found this, but the reason this was hidden so far is that it happens in a segmental duplication that is exactly that size. So we are losing a lot of information in these extremely repeat-rich regions of the human genome, and only the long-read technologies will discover those. We've now done that in collaboration with 3 other centers. We did 58 trios. These are the average numbers. Most importantly, you should keep in mind, only 1 in 4 trios harbors a de novo structural variant. Not all of them are disease-causing, but there is at least excellent candidates for further follow-up. So for now, at this very moment, it's still a bit preliminary. In 10 to 15% of cases, we have an excellent candidate as a de novo, and in 5% of extra cases, we even can look at compound heterozygous structure variants to other variants on the second allele. So I'm actually, even as the uber-optimist of the department, I'm surprised by the current success rate of such a technology that in previously heavily studied cases, we still find candidates in at least 15% or so that are absolutely worth pursuing right now. So our joint future vision of OGM in the clinic is that a cytogenetics laboratory that looks usually like that, a lot of errors, a lot of parallel workflows are needed, can be quite easily replaced by just an OGM workflow. And to achieve that, we even invested further in BioNano's technology, so we're amongst the first in the world that have their latest instrument that should arrive next week. And we also purchased another automation robot to really get this in a fully automated kind of workflow. Now switching gears to the real long-read sequencing, and I brought one story from the SolveRD consortium. So that's a European, pan-European consortium that we're co-leading that really tries to tackle the unsolved rare diseases and rare disease cases and families. So it's a huge consortium, more than 300 partners across Europe, and particularly one branch of that looks at long-read sequencing, how it can solve so far undiagnosed rare disease cases. Don't have to understand all the details at that stage. When we performed the data acquisition here in Nijmegen, we could only afford a 10-fold coverage genome with a long-read sequencing, but we collected by the European experts the most precious rare disease cases to do so. And today I have the first data of the first 300 or so samples representing 114 rare disease families. We even got together just 1.5 months ago in our Solvathon. So that's a new concept of SolveRD, bringing the data and clinical disease experts all in one room and really on the spot try to solve some of the unsolved cases. That's not only great fun, but it's also really productive to bring some of the smart people in their respective expertise together to really get going. Just as an example, we, for example, identified true disease causes in 2 patients with DMD. They just carried inversions that the previous technologies were unable to pick up that disrupts DMD, and that really fits Duchenne's muscular dystrophy really spot on in terms of the diagnosis, but missed by all previous tests. Secondly, just to give you 2 examples, there were 4 cases with suspected forms of ataxia or movement disorders, all of which carried repeat expansions that had been missed by the previous tests. And obviously the long-read technologies can find these STR expansions with relative ease. So bottom line, for such a study, even heavily pre-studied cases, we now find the disease cause in close to 11% and generate even additional candidates that deserve further follow-up. So to sum up that part, long-read sequencing, and in this case it was even modest coverage, is really finding mutations on all levels, and in particular those ones that have been hidden or missed in the previous analysis. So really good disease candidates in 11%, properly solving cases even, and candidates in many, many more, and I think the 6% is just something very conservative that is very close to a diagnosis. We had also very specific disease syndromes in here. Sad news is we didn't solve a single of those, so there's some syndromes around that look genetic, probably they are, but still, even with these latest technologies, we don't crack them immediately. Last vignette for the long-read sequencing, the data I've shown you were on the previous generation, the so-called sequel on PacBio. We invested in their latest instruments in Revio, so we have 2 of them because we like them so much. There's even more coming this year. And what we're really doing with this is checking how good those genomes are. And there's basically 2 important questions that we ask. Would this be the very first generic test for all of genetic germline testing? So can we replace a test portfolio of 20 other tests in the lab? So we're currently running the 100 most difficult mutations we've ever seen in our department. And secondly, similar to the SolveRD effort, but now much more systematically, we ask how much extra can we find in cases that underwent previous testing. And in a much more systematic way, I hope we can really provide evidence for this very soon. We are also involved in European consortia to do very similar efforts. Just to finalize the long-read stuff here, if we do these hi-fi genomes now, just showcasing some stuff of these 100 mutation-positive cases, we find mutations on all levels and also the most tricky ones. So that is deletions, even in extremely GC-rich regions. It is mobile element insertions, really a nightmare for short-read data usually. We find mutations in pseudogenes and otherwise very homologous sequences. We also find these repeat expansions, now with a higher coverage that Revio gives, even with more accuracy. And on top of DNA, we also see DNA methylation, so that's actually quite nice. We also already published first papers on this. So for example, de novo mutations, similar to the OGM, that are so tricky to find if your technology isn't accurate. Read up on the manuscript of our PhD students. It's just out since half a year. Final vignette on the long-read technologies is we can not only use that for DNA, but of course there's also intrinsic value of longer RNA sequencing. Here we did some pilot studies together with Peter Bram Thun and his team, and we looked at in vitro stimulated immune cells, PBMCs. And those are just an example of an accessible cell type where we still want to discover novel biology, and we did so by long-read sequencing. So different from anything you're used to by counting or Illumina sequencing, where you deduce the isoforms, here we can really read transcripts from start to end, so we really know the ground truth immediately. The important message here is, even if your cell type or your organ has been sequenced massively in the past, once you start using long-read isoform sequencing there's many more transcripts than we ever thought. Whether all of those will be used to produce protein is a completely different story, but that biology of transcripts is much more complex than we ever thought. There's 2 papers already out, one preprint and one published paper on different tools, so if you're interested, read those up. I'll just finish with the last minute to express the why we're actually doing that. So these long-read technologies can really stop genetic odysseys and diagnostic odysseys. And just to showcase that, I brought an example collaboration with former colleagues, Marjolein and Roland, who now work at the Princess Maxima in Utrecht. They have studied a family that had full tragedy in their family. They lost 2 of their girls, one after 2 years, the other after 2 weeks after birth, due to a terrible brain tumor that is known as ATRT. And if you have recurrence in a family, you know quite exactly what's going on. There must be a germline mutation in the disease locus that is well established in SMARCB1. Because they had the recurrence, all diagnostic testing was performed. Sanger sequencing, MLPA, CNV microarray, whole exome sequencing, and in a research basis, even whole genome sequencing, no mutation found. Then we applied actually the mix of the 2 technologies that I've showed you today, optical genome mapping and long-read sequencing by PacBio, and those finally gave us the clue. There was a mobile element insertion in the intron of SMARCB1 that all the previous tests clearly missed. If you're interested in the details, that's written up, and you can dive into that. But much more importantly, my dear colleagues in diagnostics dared to develop a prenatal test immediately in this family that had this full trauma and very negative experience of a rare disease that is so traumatic. And because of the prenatal test, the family dared to get pregnant again. The mother was only 40 years of age. That was also successful, and lucky enough, the very first and very early prenatal test discovered that the mutation was not present in the subsequent pregnancy, and since 1.5 years, they have a healthy baby boy, and were even kind enough to give us a little interview, so if you're interested, there's a video of that online. That brings me to the most important slide for those that really want to catch up with long reads and what the benefits are. Just to sum up, this gives us access to hidden mutations, in particular structure variants, repeat expansions, and some more. I think only now with these technologies we're allowed to speak about whole genome sequencing. All the previous tests were simply not whole. Hidden mutations will explain still a lot of undiagnosed rare disease. I'm really convinced by that. Instead of moving only towards digenic, multigenic, multifactorial, I think there's still a lot of hidden treasures in the genomes that we overlooked so far. So in a sense, only these technologies make human genetics much, much easier. Probably we only need one test in the future, and we really move not only from variants, which is just a proxy for all the work we do, we actually can start talking about disease alleles and disease haplotypes, even for rare diseases. I only gave you a little pitch on the RNA-seq, also there we're getting closer and closer to real biology. So ultimately, I think there will be a single generic test making our life, technology-wise, maybe a little bit boring, but for our patients and for our discoveries, much, much more exciting. With that, I'll finish by acknowledging a lot of people. Sol4D was really a pan-European effort, but even in-house, many, many colleagues were involved. Great thanks to them. We have an amazing team in Nijmegen that I'm really excited to work with, also for the years to come. In particular, I want to highlight our technology center that does a lot of the sequencing efforts that I've shown today. With that, many thanks, and looking forward to any questions and discussions today.
Thank you, Alex, for giving this very nice overview of new technologies, and especially technologies that have shown to be such a powerful technology. There's room for one more question, maybe from the audience or maybe online. No? Oh, there's one. Peter Bram.
Yeah, that was a great talk, thanks. So you already showed that all the cytogenetics can be replaced by optical genome mapping, I think, and then you continued by stating, well, maybe everything can be replaced by PacBio sequencing. Is that including all the cytogenetics that you showed? And the second question is, can you comment a little bit on the current costs and how much PacBio should go down in costs to make this happen?
Yep. So first on the OGM, so indeed, I suspect that some of the long-read sequencing technologies on the long run will also cover all the cytogenetic aberrations once it comes to germline. There may be even some exceptions there where we see that OGM with the length of the molecules, when we get 2 megabases, there's some sequencing that sequencing still can't do. So I think there's a niche for the next years, but maybe ultimately long-read sequencing may be the only test for germline. But there's a huge benefit of using OGM in cancer and in leukemia. So for just €400 reagent price, we can cover a genome up to 1,000 or 1,500-fold now. And that naturally comes with sensitivity for acquired or somatic SVs. Hence, there's a huge niche. So probably there may be a generic long-read sequencing workflow in the future just for germline, but for anything where you suspect somatic, and we could even debate for hours where you suspect that, there I think OGM is certainly very valuable on top. So I predict that for the years to come, it may exist in parallel, and even after for tumor and leukemia, it may exist longer for OGM, while long-read sequencing really may be the go-to test for germline. In terms of price, OGM currently around €400, PacBio reagent cost only. PacBio roughly 1,100 reagent price, and we calculate if you replace also all the workflows that we currently have, there's still some homework to do. I think in the Dutch system, I think once the price hits €300, we could use it potentially as the only and generic test. So we're a factor 3 to 4 off. Yeah.
Okay, I think we need to move on to the next speaker. The next speaker will be a pitch from one of our very valuable sponsors. I'll give the floor to Canopy Biosciences. Hi.
Thank you.
Oh, you don't need that. You can use the mic there.
Yeah. I guess this is the clicker. Let me see how it works. Okay. Yeah. I like my tonics straight. Let's get that first addressed. I'm working for Canopy Biosciences. I'm based in Amsterdam. Canopy Biosciences is an American company based in St. Louis. We got recently acquired by Bruker. What's interesting to know, we have 2 branches of business. One is we offer spatial services, and the other branch of business is we are making our own spatial proteomics platform. The focus of this talk will be on the platform, not on the surface. Past weekend, I was in the museum, and I was with my nose on top of this painting, and I could not really distinguish what's happening in this painting. As I was walking away half a meter, I start noticing there is a couple in the painting. And then I moved further away, and I could see it was a lovely painting of a garden with multiple couples in love. And this is a perfect metaphor of why spatial is very important. In order to capture your full biology, you need to analyze your analytes in your tissues while they are intact, so you can capture a full biology, your full biology. This painting is in Amsterdam. If someone knows the name and can guess it, come to my booth and I'll give you a pair of socks. Why is spatial so important? We know now that cells are interacting with each other. So spatial will tell us which cells are communicating with each other, especially in situations of oncology and tumor. We know there is a high communication happening between immune cells and tumor cells. And spatial allows you to capture that interaction. Our platform Cellscape was born in a flow cytometry lab in Hanover. The reason why it was a flow cytometry lab is flow cytometry, it's a gold standard technique to analyze your cells. It's very powerful. You can analyze dozens of markers. But the downside of flow cytometry is you're dissociating your tissue. And then you have another gold standard technique, which is immunohistochemistry, which is allowing to analyze your tissue while it's still intact, but the plexity is very low. So in Hanover, they decided to marry the technologies. And it was a happy marriage because then Cellscape came into existence, which is a platform which allows you to analyze dozens of protein analytes in tissues that are intact. I will now go into the workflow a bit. The technique is a very straightforward technique. It's no magic box. In the middle of the slide, you will see the Cellscape platform and also the microfluidics unit. Basically, you grab a normal histology slide and you put your FFPE tissue on it or your fixed frozen tissue on it. And then you put your antibody cocktail in the microfluidics unit. And then you can put up to 4 slides in the Cellscape platform. And the Cellscape platform executes an automated workflow where it's staining your tissue per 5 antibodies. After it stains your tissue with 5 antibodies, it images those fluorophore signals, and the signals then will get photo-bleached, and the cycle will repeat itself. It can do this in a virtually unlimited manner, so you can go up to 20-plex, 40-plex. We have even data showing 100-plex. After it's done acquiring this data, we're going through a whole data pipeline where we are using background removal so we can grab a very crisp signal. It's an open source platform. The data that's coming out of the Cellscape can be analyzed with third-party softwares such as Physioform, QPath, and you name it. Since we are an imaging-based technology, I will demonstrate a data set and walk you through it, how it works. Here you see a colon sample which has been stained with one of our 21-plex panels. And you create a high-resolution image, and you use the DAPI staining that helps you to do nice cell segmentation. Once you have the cell segmentation, you're able to quantify where your signal is coming from, and you're able to cluster your cells, and you're able to analyze specific subsets of cells while keeping spatial context. So you're able to really capture these rare subcells in your data set and pinpoint their X, Y coordinates. And you can, for example, analyze these immune cells which are close to a tumor and understand their relationship with each other. The platform has been validated on dozens of tissues and hundreds of antibodies. It's compatible on FFPE, fixed frozen, PBMCs, and all kind of different species, such as mouse and human, non-human primate. One of the major strengths of the platform is it's compatible with open source antibodies. So you're basically able to use your antibodies that are in your lab. You don't need to pay expensive prices to vendors to validate and customize your assay. You can do it yourself. But next to that, you can also use one of our ready-to-use panels, which we have developed multiple ones for FFPE, fixed frozen, PBMCs, and you name it. A few months ago, or a month ago, we had a very exciting announcement. Namely, CellScape is now compatible with normal histology slides, which is a very big development for the field, because majority of the labs are working with histology slides. And this effectively allows us to image close to 710 square millimeters. This is massive, because you're able to do spatial proteomics integration of your tissues in a very high throughput manner, and you're able to analyze TMAs ranging from 50, maybe, depending on your skills, maybe even 100 per slide. And the instrument allows you to analyze 4 of these slides simultaneously. And next to that, we are reaching a nice resolution of 180 nanometer, so you're at the subcellular level. And this concludes my presentation. If you want to hear more, you can visit me at my booth. If you want to win the socks, then be quick. I think I saw a few people already looking at me. I think they probably know the painting. It's a Dutch painter. Just FYI. Thank you, everybody.
Okay, doesn't work? Yeah, now it works. Any questions? Maybe? We have a quick question. I think we have time for one quick question. No? Well, you know where to find him, at his booth. So the next speaker is Danique van Rijswijck. Oh, the chocolate. That chocolate. Don't forget the chocolate. Yes. Danique van Rijswijck, we don't know each other. I don't think we ever had a gin tonic together now, but what I know is that she's going to talk about the characterization of immunoglobulins, and as a pharmacist, I can tell you that this is a very important and timely topic. So the floor is yours, Danique.
Thank you very much, and thanks also to the organization for giving me this opportunity to talk here about our work, which is about the immunoglobulomics, which we see as the in-depth characterization of immunoglobulins from human biofluids. So first of all, I want to give you a little introduction into antibodies, because my whole talk will be about antibodies, so it will be important that we are all on the same page. So let's start with the B cells, because actually these B cells already express a membrane-bound antibody, also known as the B cell receptor, or BCR. And once this BCR binds to its cognate antigen, the B cell gets activated, and it can either differentiate into a memory B cell, which is there for long-term protection, or it can also differentiate into a plasma cell. And this plasma cell is then responsible for secreting the BCRs in the form of immunoglobulins, or antibodies. And these antibodies can then in the blood neutralize your antigens. So we see that these antibodies are really important for neutralizing, but also for long-term protection, as you see that we get these memory B cells, but also the long-lived plasma cells. And also the clinic saw these nice advantages of these antibodies, and now they're also used often as monoclonal antibodies as a therapy in clinic. And if we then zoom in and look at the structure of such an antibody, then we can see that it exists on an Fc tail, which is more constant, with 2 Fab arms. And if we then continue with looking into that, then we see that these Fab arms are important for the antigen binding, and therefore need to be really specific, and therefore also vary a lot across these antibodies. While the Fc is way more constant and is important for effector functions, like binding to the complement system, and this is way more constant in the fact that it comes in different isotypes, but within these isotypes you have different subclasses, but that is it. It's not more differentiated than that. And IgG1 is the most abundant antibody subclass that we have. And normally these antibodies are selected to bind to outside pathogens and thereby neutralize your pathogen, but this can also go wrong and then you develop an autoimmune disease. But let's first focus on these Fabs, because these Fabs, as I said, they need to be really specific to bind to these antigens. And to do that, it also needs to be really specific, and it varies a lot across these antibodies. And if we zoom into this region, then we see that it actually consists about different complementary determining regions, or CDRs, and they differentiate that lot because they are either differentiate because they are formed by VDG gene recombination, and thereby you can have different genes that come together, and thereby you can get combinatorial diversity, but also in these junctions where these genes come together, you get even a higher level of diversity called junctional diversity. And once such a BCR is activated, it can either undergo another round of affinity maturation by somatic hypermutation. As you may appreciate, you can get a lot of these different Fabs and a lot of these different antibodies, and all these antibodies will then, in the end, make up your antibody repertoire. So actually your blood and your circulation is filled with all kinds of different antibodies. And if we then have a zoom in and look at the current techniques that are available to look into these antibodies, then we can differentiate that a bit along 2 sides. So first of all, we have techniques that focus on these BCRs. So let's say, for example, the B-cell sequencing techniques. So they are now able to sequence these B cells, BCRs that are present on the B cells. But as you also may appreciate, these are not real antibodies that enter the bloodstream or the circulation. And also we don't get any information about the abundances. Then there is another part of the techniques that is focusing on these secreted antibodies. Let's say ELISAs, but also plasma proteomics. But they are also focusing on they are looking at the total levels of this antibody repertoire, and thereby not looking into the characterization of these different clones that we got with the differentiation. And this is also partly due to, because it's difficult, because there are so many heterogeneity because of these sequence variants in the Fabs, making it really difficult, but also the FC is often glycosylated, making it even more diverse. And also the size, it's quite big, it's 150 kilodaltons, so therefore it's also most of the time difficult to study. So there's a need to study these circulating antibodies in clonal detail to really differentiate these clones from each other. And that's where we developed this technique for, and we developed a method to actually differentiate these clones from each other. We call it clonal profiling, and how we do it is that we start with a blood sample of patients. We then capture all the IgGs from this, so we already make it less diverse by looking at the IgGs. Then we use a specific enzyme, in this case IgDE, and that's specific for IgG1, so make even more focusing. And with that, we look at the Fabs. So the FC sticks on the bead, and the Fabs we can then analyze. And as the FC sticks on the bead, we also get rid of the glycosylation that's part of the FC. And with that, we can then analyze these Fabs. And if we then look what we then get, then we do it with LCMS. So you get for those who are not familiar with that, you get just a time trace, and then you get different peaks. And actually, if we look at these peaks, then we see that these different peaks are different Fabs. So although they are quite similar, we can differentiate them with LC-MS. And then if we deconvolute them, we can either say, okay, we get a nice spectrum of all different peaks with different abundances, and we call that our FAB repertoire. And also we can even more differentiate it because all of these peaks have their unique retention time. So we say that a unique clone is a unique mass plus a unique retention time. And yeah, with spiking in monoclonal antibodies in our blood, we do that on site. And we know the abundances of these monoclonal antibodies. We can even quantify them and even give them, you see on the y-axis, the concentration, or the estimated concentration. And this is quite smart, and this is just, I think, a nice example of what shows what our method can do with the resolution I showed you. Is that we, for example, this was in the really beginning of COVID in Italy. They didn't know what to do, so they just gave them a lot of monoclonal antibodies. For example, tocilizumab, which is an IgG1 monoclonal. Yeah, and we got the plasma profiles of these patients. We did our trick, so we did our method, and then we again get this FAB clonal repertoire. And what you then see is that one clone is really popping out. And actually we then saw that this is the monoclonal antibody, tocilizumab, that was spiked in these patients. And then we got also 3 time points from these patients, and you can follow, or more time points actually, but you can follow the monoclonal antibody over time. So we could even give it the half-life of this antibody. I think this is just a cool thing that we can do with this method. But okay, we did this on a healthy donor sample, and then we saw that these IgG1 repertoires were actually quite simple in the sense that we could actually separate those FABs. They were unique, so across different patients we didn't see any overlap, and they were dynamic. We see clones popping up, we see clones going down. But then the big question came up, like, okay, this is all great, but against what target are these clones directed? So we even expanded our method that I just showed to you a little bit further by diving into antigen-specific repertoires. So then up front of the whole method that I just showed you, we then also capture the antibodies specific to a specific target. In this case, because SARS-CoV-2 was still happening, or COVID was still happening, we also, let's have a look into the SARS-CoV-2 specific repertoire. So that's what we did, and then you see, as you may appreciate, you get a full plasma repertoire, you bind it, you also get an antigen-specific repertoire, and you see that they are quite different, and that you also really needed to zoom in to come to this antigen-specific repertoire. Well, as at that point also different variants were still emerging, we also had a look into the different panel of SARS-CoV-2 variants, so the wild type, the alpha, the beta, and the gamma. And did this all in the same manner. And then we could compare all the different clonal profiles that we could see across these different antigen-specific repertoires. And then we could also follow the clones and see if they were cross-reactive across the different variants or not. And then you can also see that some of these clones show a nice cross-binding towards the different antigens. For example, the one with the yellow dot. But you see, for example, also this brown dot, it's really a well binder towards the wild type and the gamma, but it doesn't bind to the alpha and the beta variant. And this is for another patient, a little bit a different visualization, but here every bar is one unique clone. And you see, for example, that this is a patient that's infected with the wild type variant. You see that there's one clone really popping up that's 10 times higher as the second most abundant clone against the wild type variant. Here the yellow clone, or the yellow bar. And then if we follow it across the different antigens, we actually see that it's not the best binder towards all, the most cross-reactive. So then we also say, well, for example, this blue one, you see it's not the best binder, but it's the most cross-reactive one. So actually the most abundant one is not always the best one to also, for example, use in clinic as a monoclonal antibody therapy. And then we were also questioning, like, okay, great, are these antigen-specific repertoires still unique for each patient? Yes, they are. What we see in this heat map is that within one patient we see the dark red spots, so they are really similar to each other. We see a lot of cross-reactivity, while across the different donors we don't see that. And we also see a lot of differences between the donors, and we saw that, the donors that got most severely infected, that they just had the highest level of antibodies, and that therefore also the most cross-reactive antibodies were happening there. So now I want to switch gears a bit with you, because as I thought, these were all antibodies in function. But as I told in the beginning, you also have sometimes antibodies that are in dysfunction, so they are binding towards your own body, and that's something that you don't want. And an example of that is rheumatoid arthritis, which is an autoimmune disease. And these autoantibodies are then made by autoreactive B cells. And in rheumatoid arthritis, one of the most well-known autoantibodies are ACPAs, or anticytolytic protein antibodies. And it was already shown in literature that the levels of ACPAs were correlated with the disease activity. So we thought, okay, let's do our similar approach on these ACPAs. So what we did, we started off with RA patients. We looked into the ACPA-specific repertoires and also looked into the total repertoires. And what you then may appreciate is that these ACPA repertoires are actually also super polyclonal. We see a lot of different peaks. And let me see. What you see is that while the plasma repertoire is focused around 48 kilodaltons, so these FABs, the ACPA repertoire is actually a bit higher in mass. And this was not only for this one patient, but we looked at different patients. And as you may see here, it's a density plot. Then you really see that for all these patients, this was true. And actually what's nice from our resolution, we can even look into single sugar moieties, and we can even find back different peaks that belong together based on these sugar moieties. So actually also different peaks in this FAB profile also belong to the same clone. So, and then we saw that this was already known, and also it was already reported that these ACPAs were highly glycosylated, so this is also the mass shift that we see. This was in previous literature, they already said that around 100% of these FABs were glycosylated, while if we look into it, we see around 60% of the ACPAs are FAB glycosylated. But then with our technique, we can go even further, and we can also look, okay, is it then one FAB, or one glycan, or 2 glycans? So we then even see that our big proportion is also carrying 2 glycans on one FAB arm. So then to conclude, our FAB clonal profiling is great to study the IGs in clonal detail, and this is information that's often missed by doing plasma proteomics or ELISAs. We see that every person's antibody repertoire is really unique, and it's also diverse. For the SARS-CoV-2, we saw that not always the most abundant one is the most informative one. It's not the most cross-reactive one. In rheumatoid arthritis, we saw that the ACPA response is polyclonal, and we also saw that it's highly glycosylated with having 1 or 2 glycans on the arm. And then I want to also to give you a little summary and a perspectives. So while I showed you here IgG1 repertoires from blood on the total and on the antigen specific, we are now also expanding it towards different subtypes. And also we are looking into different fluids, for example, urine or milk, and see how they compare with the plasma repertoire. We are also diving a bit deeper because now we see the FAB mass. We are also working a lot on really sequencing or getting sequence tags to see from which genes they are originating. And with that, I think we are further challenging the immunology and rewriting the textbooks. With that, I want to give a big thanks to my supervisors, Albert Bond and Albert Heck, and also the whole team from the Heck Lab who is working on these antibodies, the LUMC for the collaboration about rheumatoid arthritis, and the AMC and the RIVM for the nice collaboration about SARS-CoV-2. With that, I want to thank you all for listening, and if you have any questions.
Now, thanks for the inspiring talks, showing that digging more into the detail can actually deliver novel biology. Any questions from the audience? Now then, I have one quick question. Where do you see this clinically applied? So where else than in rheumatoid arthritis could that help in clinical decision making?
I think if you have just antibodies that are really specific for different diseases, you can track them. But also, if you want to see like monoclonal antibody therapy getting developed, we can now really see, okay, which antibody is the best to go further into that. So I think in that stage, because now it's often relied on these B-cell sequencing techniques, which is nice, but yeah, it's not telling the ground truth of what is really the most important antibody. So I think we can help patients with that.
Thanks. Don't forget the chocolate.
Thank you.
Thanks again. Okay, we have one more pitch talk from SomaLogic before we go over to the visioning session, which will be guided by Alain. Hello.
Hello. Just take this one. Right.
Everything set up?
Yep.
Okay.
Sounds
well,
good. Thank you.
SomaLogic, the floor is yours.
And I just press this.
I guess so.
Super. Hello, everyone. So I am Eshita Mutt from SomaLogic, and here today to talk about the power of proteomics. So even though I am really enjoying the lineup here and I'm seeing a lot of proteomics talks, I'd like to talk a bit about how SomaLogic is a little bit different to that. So why proteins? Of course, we all know that more than 95% of the drug targets are proteins. And that's why it makes it very important for us to know what the proteins are doing. In the same way as you see the picture above, where it is showing the butterflies and the caterpillars. So both of them have the same genotype, but what is different is the protein set or the protein expression that is going on. Now, even in a disease pathology, this is the same thing that happens. You start off from a normally healthy person, and the set of expressions changes, or the proteins changes. And you might be interested to know, okay, which proteins are they? If you already know them, okay, you go to the therapeutic part of it, go to a clinical and get it tested. What if you don't know? If you don't, then the obvious choice will be to go and assay all of them, or the ones at least which are technologically possible for you to do. Now, SomaScan assay actually helps you to do that with all the repertoire. So as you can see in the assay here, we started off in 2000 where we were developing our technology, and I'll tell you about it in a bit. But you can also see that we have launched the 1000 plex we started off with, and now from 7,000 to 11,000 plex. So thinking about it, you can measure 11,000 proteins at one go from one particular sample. And that is almost half of the human encoded proteome. Now, what is also crucial here is that this has been widely adapted. It has been used across various multiple matrices. Of course, some of them very complex, like plasma and serum. It can also be seen across the different citations and the publications that has been present. Now, SomaLogic's SomaScan assay and also other products that we have, like the SomaSignal tests, these also can help you across the whole drug development pipeline. So either if you're working at the research phase, you're working at the preclinical, you are at the population screening, or at the diagnostic part of it. So for all individual parts, we can help you tailor your work presently. Now, one of the parts mentioned here is the biomarker discovery. In any type of, let's say, if we are taking the example of a plasma biomatrix, you all know that some of the challenges that comes for biomarker discovery in that is, let's say, for example, the low concentration of proteins or the biomarkers that is present, or else sometimes also let's say your biomarker is actually blinded by the highly dominant proteins like albumin, which is very easily sometimes they saturate or oversaturate your assay. In case of SomaScan, we have been able to overcome and measure both the low abundance and the high abundance protein with high specificity. Now, let's meet the star of the show. So that is a SomaMer, that we call it a SomaMer reagent. So it's actually a modified aptamer. So when I say aptamer, think about a single-stranded DNA, which is forming a 3D structure and doing like a selective handshake on this particular protein. So here the protein is the one in gray, the SomaMer is the part in green. The parts which is in purple are actually the hydrophobic patches where we do some modifications to that particular single-stranded DNA, and that helps to increase the specificity of the whole somamer. Now, due to this high specificity, we have been able to scale it to very high levels, as you saw before, and also it has a specificity comparable to monoclonal antibody. Another point that I would like to mention here is about the reproducibility. Because it is based on the SomaLogic technology, the batch-to-batch variations is hardly there. So that is also something which is very useful when we are working on a longitudinal kind of data set and you would like to combine the data sets together and you are thinking about how to overcome the technical noise. So this is one of the reasons. Now, you can ask me that, okay, so we have our antibodies, why go for somamers? So specifically monoclonal antibodies. So this is a very some of the points to show that yes, antibodies are there and it's very prevalent. But we also have to take in mind that monoclonal antibodies are expensive and also takes a long time to cultivate. Whereas somamers are synthetically produced, so they have the batch-to-batch variations is hardly there, and you can scale it up also at a very high level. If you're thinking about polyclonal antibodies, which are cheaper to produce, you also have to keep in your mind about the variability that is present there. Now, let's talk a little bit about the different applications that you can do with SomaScan. Of course, I don't have all the time here to talk about all of them to you. So I will tell you about 3 different applications which we worked across non-human, that is mice, human, and also different big cohorts. Now, a little game here, if you all will be interested, is that among these 3 case studies, I would like for you to find out how many number of proteins or which case had the highest number of proteins measured. Yeah? And then you can come and tell me at my booth about that. Let's see. So have you ever heard of Mighty Mice, the one which went to space? Can I have a show of hands to know who all have heard about Mighty Mice? Okay, so this was a knockout mice which was made from the by knocking out the myostatin signaling pathway. So the interesting part is this particular mouse is now being used in a scientific study to find out muscle and bone loss and how it can be treated or how it can be stopped. Muscle and bone loss is a very big issue which happens in all of, in any prolonged space flights for astronauts. And even after they come back and if they recover for 2 months or so, sometimes bone loss is so much that you cannot get back to it. So this particular study, so what they did was they were targeting the myostatin and activin pathway, and they did that by checking using our SomaScan assay, and they were doing that across both the space-bound and the earth-bound mice. And after that, they were able to find the differential expression across, and they sorted it across the top high-regulated and the down-regulated proteins, and they found that these were actually impacting the bone, the homeostasis, and also the bone and the muscle mass regeneration pathways. Now, let's go to the next slide. So this is about aging. Now, of course, all of us are interested in how we age. Can we tell something when we are aging? Can we tell, okay, I'm going to get this disease soon or not? Yes, genetics can do that to a certain extent, but let me tell you why proteomics can also do it a little bit better also. This particular research group, they looked at the different plasma proteomes across various cohorts. So they looked at very big cohorts of only older individuals and also of across all ages. Now, what they found out was that they coined something like a proteomic clock, and they were able to find that how your protein changes across waves in 3 different decades of your life. And that also kind of gives you an idea about how or what kind of diseases you might be susceptible to. And you can monitor that also using proteomics. This group further went on to actually detect the age of different organs that you have. So in the sense, like, even if you might be in your 3rd or 4th decade of life, it might be that your organs are much younger or much older than what your actual birthday is. So they were able to find the aging across 11 major organs, and they were also able to see that if the organ aging is imbalanced, then that can be quite, it will increase your mortality by 20 to 50%. One of the examples given here is how if you have your heart age is like very high, then you can lead to 250% increase in your heart failure risk. One of the strengths of the platform that I would like to show here is present on the top, like how the globally correlated data and the low CV helped, because we were working across 6,000 samples, which is spread across 5 different cohorts. And the last one is this different sample matrices. This was a study done on Alzheimer's disease where they were working with, you might know that Alzheimer's is a very heterogeneous disease. So it is very difficult to actually divide or find out which particular symptoms or which particular proteins are related at which step of that. So and that's why many times scientists actually go and work only with one matrix. So they work with only CSF or maybe some of the plasma. This particular study was working multi-tissue proteomic profile across 2 different cohorts, and here they were able to find protein changes and able to detect it using a model to find the differences between the sporadic Alzheimer's and the healthy individuals. So that kind of shows you why SomaLogic has a different way applications across different matrices, but also across the different research the drug development field. So these are some of the value propositions and the strengths of the of our platform that we saw. And just to finish off with, I hope you caught, I did give you some hints as which of the case study had most of the proteins detected. So please come and tell me, and we'll be happy to come and answer any of your questions or also listen to your research. Thank you.
Thanks for this nice talk. I think for the sake of time, we have to move on. Please visit the booth if you have questions, and Ala is going to take over the visioning section or session.
Yeah, thank you, Martin. So thanks, Marcel and Martin. Marcel will stay around, so
yep.
Can I have on stage here the core team of the X-omics and Eric Reits and Peter Horvatovich. Eric and Peter will introduce themselves, but we need your help here because we haven't done this before, but it's also fun to do things at a meeting which we haven't done before. And in this case, I'm going to shortly introduce the reason why we're having this session, but we'd like to have your input here. So we call this a visioning session. Visioning session, some people ask me, like, what do you want to do? Well, basically it's a brainstorm with like 250 people here, it's about 50 people online currently. So we're going to do a brainstorm. And the brainstorm is about what is the future of spatial omics. You've seen several displays of technologies, great things which are happening, but it's going at a pace that we cannot predict. So I'm going to ask you in a few moments, where do you think we will be in 5 to 10 years' time? So let's see how it is. The background of this is the National Roadmap on Large-Scale Research Infrastructures. And several people from NWO are here who made this fantastic journey with us to help organize the technology infrastructure in the Netherlands. It started in 2015, and we had 2 rounds of this, and the funding of the X-omics initiative is part of this funding on the National Roadmap. So if you visit this website, you can read much more about it. We did this exercise a few years ago again, like mapping what kind of high-end research infrastructures are there in the Netherlands, and it was grouped by a committee into 9 different groups. And the groups are separated in the applications here of the technical and nature sciences, life and medical sciences, and social sciences. So all the lineup, so this PDF is online as well for you to it's interesting material to read. And all of these groups actually have a number of infrastructures associated. So the CrossOmics is part of this Life Science and Enabling Technologies group, and Eric and myself, together with Mark Baldus, an NMR expert in Utrecht, try to coordinate this group. And we had to define a mini roadmap. Where do we want to go to in the next couple of years? So this mini roadmap was designed after lots of brainstorm of all these experts from 16 large facilities into this kind of schedule, which we're graphical people, so we like to visualize things. So we plotted all the expertises which we have here, tools, technologies, different expertises, and we ended up in grouping molecular tools, omics, imaging, and translational models, human interactions, etc. And we have 2 main axes. One is actually to bridge the molecule to the organism. It's great to study a single cell, but it's as important to study a whole individual or a population of people, but also to move from a technology and to bridge it to the application. And there's value in all of these. And our strategic approach is to integrate those tools with omics imaging to translate towards human and other models. And one thing we had here is a vision. Where do we want to be in spatial omics? There we combine all these different things. And we had some pretty good thoughts, we thought. And it started with some first thoughts, and we'd like to have this brainstorm here. And Eric, then it's up to you.
Yeah, can we switch the computer to the other one? Yeah, it should be me. Thanks, Eric Reits. I'm chair of the animal bioimaging, so the counterpart, I would say, of the CrossOmics.
Use the mic.
Together we go for the CrossOmics. So the animal bioimaging, it got financed last year, so 25 million euros, of which 15 million from NWO. It's a fantastic team, I would say, because especially by microscopy, it includes all the universities, academic hospitals, but also thematic institutes. So you have a lot of experts here. I think Sylvia is already seen here on the left, already mentioned early on, Sylvia over there, the high content screening. Also Katie Wollstonecroft, second column at the top. This huge effort for the data management, to link all these universities, all these core facilities together for the data management. So a lot of experts together in this dream team. Because we have to address a number of challenges. All these instruments are in core facilities. We will enable scientific breakthroughs by so many life scientists, but only together as this consortium, we can address particular challenges together. Just a few examples. For example, probes. There are teams in there that also for the life scientists, they can develop probes, all kind of tools. Also important when we go for visual omics. That you can think of probes with click chemistry. You can look at living cells and do a trick, and they can hopefully combine it with an omics technology. 3D, organoids. This is an example. We saw this morning already a nice movie of this T cell kissing with the target cell. But of course, you have to find this interaction first. And imagine you have a patient, you isolate the tumor. Here in yellow, you see tumor cells of a patient. You isolate the T cells, you modify the T cells, and you put them together. So you have 10,000s of T cells interacting with the tumor cells. Most of the T cells won't do anything at all, but here all of a sudden we have a serial killer. It really starts killing all these individual tumor cells, and it goes to the next one. So this is fantastic. A T cell that is now killing tumor cells. What we can do right now, we can do the imaging. Afterwards we do the analysis. You can see it on the right, the tracking. Is it just kissing? Is it sniffling? Is it dying? Or is it really killing? But what we want to do is on-the-fly analysis, so that the microscope itself, it is tracking these T cells, it's identified as superkiller, and then with smart microscopy, you can design a tool, isolate this particular T cell, and go to the omics. This is one of the moonshots later on, combining microscopy with omics, 3D, in time, and then linking it to omics. High content screening, already mentioned with Sylvia, and also at the NKI, and again, combining also different probes, tools, CRISPRs. Again, accessible infrastructure, in core facilities, so open for all the life scientists. Correlating, of course, with correlative microscopy, 3D models, and in vivo models. And again here, the smart microscopy. Again, also here, by having such a team as NL Bioimaging, you can work together with the companies because they have to make their instrument accessible for our scripts. If you do this as a single PI or as a core facility, they might listen. But if you do it at the national level, we see it with Nigen, Zeiss, Leica, they have now their instruments to make it accessible for our scripts so that we can steer our microscopes. The PhD students, you write the scripts, the students go home, they can watch Netflix because the microscope takes over, it's doing the imaging and also the next steps. And again, as said, fair data, combining, using and reusing existing scripts. There's so many scripts for analysis, but you want to reuse them. Again, in core facilities, if you get a user, there might already be a nice script for the analysis somewhere developed that you can adapt it and use it for your own user. And of course, lots of stuff, training the trainers, that we train all the core facility staff members, and yeah, as I said at the beginning, enable these scientific breakthroughs. We had the kickoff at the end of last year, beautiful, because microscopists are funny people like me. Ideally, I sit all alone in a dark room by myself, and now you have to work together. So you have this big team. That was nice, having them together, but also that they were presenting each other achievements, so the techniques that were ongoing in other labs. And that is ideally what you have, that you'll work together with the NL Bioimaging, and now, of course, I'm bridging it now with the omics.
All right. I would like to introduce you some aspects and technologies of spatial multi-omics technologies. And starting with the most principal way of performing molecular imaging. So many of these spatial omics technologies generally start with identifying the sample, generally with a location, 3D location in the body, and then a small sample is taken, like a biopsy, and this biopsy is generally sliced. Of course, not all the technology is doing the same. You can have also imaging technologies that is directly going to the 3D part of the body, but this is just the most common way of doing it. Once you have a slice, right, then with some imaging technologies or sampling technologies, you go for special location of that slice. And basically on this location, which can be represented as a pixel or as an area, then you get a molecular profile. And this can be mass spec based, so small molecules, metabolite, drugs. It can be mRNA expression, or some other type of readout, like fluorescent readout that you can link to targeted or untargeted molecular profile, or for the site of, you get mass of your methyl compound. And then the obtained information, based on the spatial information, you can reconstruct to get spatial marker images. So if you look at the different key aspects of spatial multi-omics technologies, I call it analytical technology which provides spatial omics, transcriptomics, proteomics, or metabolomics profile, which can be linked to anatomical structures, cells, or subcell location. You can define the 3 omics layer, which I put here, the big ones, transcript, proteins, and metabolite, but there's also many others which you can find on the side. Then the molecular approach, if it's targeted or untargeted, we have already seen today many of these technologies presented. Then how you obtain the spatial coordinates, either based on imaging technology, like microscopy, or microsampling technique, like laser microdissection, plasma capillary acquisition, or picosecond laser use. And laser-based, like what is used in mass spectrometry technique, like MALDI or laser ablation. Then you have the readout, the analytical platform, again, microscopy, so it's imaging-based technology. You can have, if you only label your compound with spatial coordinates, you can use next generation sequencing technologies or mass spec. The bioinformatics aspect or the spatial aspect, you can assess and integrate into the data interpretation in many different ways. Here I take one example from a review that is showing also the program that is used to detect spatial pattern, for example, cell-cell interaction, cell segmentation, and detect different cell types, subcellular structures, and also integration of multiomics. And finally, what are the other aspects, which I already mentioned some, is spatial resolution that you can have in 3D and 2D spatial coordinates. Dynamic aspects, it can include also dynamic aspects in this, and live imaging, that one other aspect to consider. In the sample isolation method, yeah, how you frequently and where you are sampling, this also determines which type of data you are collecting. Targeted versus untargeted, right? If it's targeted, you only look what you want to look for. Untargeted, you can go in discovery mode. The information richness of the molecular information, like for protein, the protein forms, PTNs, or sequence variants. And if you go for spatial omics, you can also integrate that with more deep molecular profile, like integration of spatial omics with single cell integration, and of course all the bioinformatics and data management is going with that.
All right, so if you click one further, so what we're going to do now is actually ask your input as well. And again, I'm joined here by the core team, so you know probably the people, but Albert Heck, Marcel Nelen, Thomas Hankema, and Peter Bramadhoen. And together with our co-leads, we try to organize the activities of the CrossOmics initiative. I've asked Janneke to monitor the chat, and I've asked Alex to walk around with a mic to solicit responses. And so we have one mic here, and we have one mic for the room. And first I'm going to ask, starting from Albert, just with a few words, like where do you see our ambitions in spatial omics in the next 5 to 10 years? So not what we're doing now, but what we're going to do. Albert.
Okay, yeah, a bit biased, but I think, you know, it's very natural that imaging becomes more molecular, and if you can detect more and more different molecules, it becomes spatial omics, because then you try to see all molecules. So I think that's really going to be the future. Going back to my roots, which is more in proteins and proteomics, I really think that what we really need is also techniques to look at the proteoforms. You know, I'm just baffled by a boring protein in our plasma, like a histidine-rich glycoprotein. I estimate that there are maybe, you know, I think at least 10,000 variants of this molecule. So I don't know how SomaLogic makes one aptamer to detect it. I think we need maybe 10,000 different aptamers to detect it. So this is, I think, the missing link. We want to see, and for me it's a very basic question, what is a protein? And for me, that is a gene product that has a function that is different than the other gene product. So if a proteoform has a distinct function, we should image this as well. And yeah, where we want to contribute, I think, is to see if we can make this spatial omics not only spatially highly resolved, but also molecularly very highly resolved. And then I mean these proteoforms, and this is maybe where I want to keep it.
Spatial molecular characterization of proteoforms of proteins. That's a really good one. Marcel.
So I am also a little bit biased, of course. I'm a sequencing buff, so for me, sequencing will probably be very important for the next coming years, especially in transcriptomics, I think, and that in combination with the spatial technologies that are currently developed. I think it will be important to get the resolution as high as an exome, or maybe even a bit further. Also methylation might be very important for the next coming years to really develop in a spatial technology. This afternoon we will see a nice example of characterization of cells and tumors with using methylation as a technology within the spatial context.
So that, I think, is for the coming years quite important and probably needs a focus from the technology development.
Okay, so that's spatial transcriptomics, but where shall we be in 5 to 10 years? Because what you're describing now is probably 2 years from here. No, I'm kidding.
Well, no,
Back in 2017, we actually said at 1 hour proteome, that will be so cool. And now we can do it like in 45 minutes. So we're really looking for those moonshots like that.
I'd like to look at 2 years already. That's
no.
quite important. I can remember a presentation that Professor Brunner gave in our department in Nijmegen a couple of years ago, where he showed that predicting is actually very dangerous because most predictions do not really become true. But overall, I think the technology needs to be fully developed to be able to characterize on a single cell level the interaction between cells within the spatial context.
And spatial transcriptomics, Eric, as well. Yeah. Thomas, what do you think? You're biased as well, right?
Correct. But let's say, so predicting is maybe difficult, but maybe more wishing, because I believe in the power of measuring blood for diagnostics and so on, but not for understanding. No, I'm afraid that we are a little bit more boring than Albert. I don't think that each molecule will occur in 10,000 different forms, but maybe a little bit more with modifications than we think. But what we have to understand, how these small molecules, which are funny enough, more or less often similar in different contexts, how they really work locally to do different things and trigger different things, although they are partly the same. So how can that happen? So the environment is very critical, cell-cell interactions are very critical, and the whole, let's say, concert of different cells being in action. So therefore, what Eric said, the marriage of imaging and molecular, deep, mass spec-based things, for example, work with Sylvia on, she has beautiful images, but can't tell what all the molecules will be in that nice pictures in all the different pixels. So trying to merge both live with maybe feedback and even modifying experiments during the measurements would be great. Do we have the technology? No, not at all. Are the big companies working on that? Well, I don't think so really, because some of these very innovative technologies which we need are maybe even not completely there yet. And there, I think, is also our niche, where we have the history of really doing microfluidic-based single-cell or subcellular-based enrichment, modification, modeling molecules in the liquid, in the gas phase. Well, that's, I think, where I see the challenges, but also the fun to work with. And again, then we as the Netherlands are rather innovative in this kind of technologies so far. So let's keep that spirit and surprise the big companies what we can do.
You're moving from a view to technologies to pitching the Netherlands, but that's okay. Peter Bram, what are your thoughts on this?
Yeah, so my bias is actually from the conference I was participating in last week. That was a conference on myotonic dystrophy. And there was a researcher from the Friedrich Bauer Institute in Munich. They call themselves the FBI because they do a kind of forensics. So they did single nucleotide RNA sequencing and identified this cluster of cells. They call it cluster X. And that actually only existed in patients with myotonic dystrophy, and they had no clue what they are doing. And these cells had characteristics of all type of other cells, but they simply don't know the function. So they also did histology, and then in the end, I think with spatial techniques and coupling to the single nucleus RNA sequencing, you can actually trace back which of these fibers they come from, but then still we don't understand the full picture. So where it needs to go is that also the dynamics comes into this. So I think we expect, or we can hypothesize, that the muscle is being repaired and dying muscles are being repaired by influx of nuclei, but how that actually works and whether we can monitor that real time with bioimaging techniques, I think that would be a great challenge.
So dynamics.
It's nice, so far, really great. You're in the floor, this is one stage higher, but you do have to earn your lunch. The ceiling. Where do we want to be in 5 to 10 years? Because we are coordinating this group, we are making plans for the next 2 to 5 and maybe even a 10 years time. So look at the ceiling. What are really the moonshots? 5 to 10 years.
That's great setup. Thanks. Thanks. I have a question. Magdalena Harakalova, UMC Utrecht. I'd like to ask you guys, what do you think about the current state of our biobanks? Because it starts, of course, with a tissue or sample to do a spatial omics on. Are we prepared for the 5 to 10 years? And if not, what needs to be changed?
Okay, I'd like to refer that because it's not a question to us, but you're stating that the ambition should be, the biobanks should be, what?
They should be prepared, because they are usually prepared from techniques that we had from
Okay. So let me repeat because of the mic. So the biobanks, ambition is that the biobanks and all the materials plus the data associated should be ready to accommodate all the spatial omics technologies. I'm going to ask you, what is needed from now actually to make that happen? Yeah, you're not getting off that easy and you need to earn your lunch. So
you are asking what's my take on that?
Yeah.
Or, so my experience thus far is that they are not, but I may be coming from, it's maybe specific to my field, but first of all, the people who are running the repositories on biobanks are not ready for the type of collection that's needed. Everything is maybe limited to 4 or 6 micron slices and maybe some bulk isolation for RNA. But the protocols in those large biobanks are not there if people want to do the spatial omics, so then it's more of a research. I rather was interested to hear what you guys think about it.
Well, if I can answer that and then ask someone else somewhere. The biobanks actually, I think they may not be ready, but they're gearing up to be ready. And for one single person like Peter Bramstas, you need multiple samples to do the multi-analysis and then integrate it. So for one individual should have multiple samples and flows and longitudinal. Then we probably get to this stage. But hey, we're not there yet. The informed opt-out is a really good part as well. Alex.
Another. Hi, thank you for being here, for your introduction. I was wondering, like, if we're talking about the future, like we've already heard about a lot about like advancements in technology, but I was also thinking, like, for instance, in 5 to 10 years from now, PhD students may be assistant professors, like assistant professors may be full professors, and on the other hand, people who are PhD students by then are still now, for instance, in high school or maybe even elementary school. So what do we need to do, like, to also get the right people in the right place and to keep academia an attractive environment to work in, so we also have the people to make these developments?
That's a good one. I'm sure any of the wise gentlemen would like to elicit there. Please.
Well, I think this is a very big challenge and goes also a little bit in politics. I think, let's say, the entrepreneurial spirit of researchers is very important, because if you want to change the world, you have to have the ideas, but also a model where you want to go to. Some people make it the difference between science and engineering, but I think there is not such a big difference. It's more, let's say, looking maybe a different challenge in a different way. But to go to schools and to explain children, I have 3 of them, how important it is to be innovative and to go, let's say, to push technologies, and then, to push it also in the Netherlands, or at least in Europe, and not just buying it from the US or from China or from other companies, but that it is great to be innovative, and that it is fun to be innovative, and to or run an academic group or start a company on something which is not yet there, but you make the big difference. I think that's the spirit which we have to get out.
And if I may add, start at the bachelor. We see that the bachelor already, they get so intrigued if we present what we're doing here already in the 1st or 2nd year. It's fantastic. But also don't forget the applied universities. There are lots of people there in the biotechnology. They will write the scripts. They will do all these instruments, because the scientists might move on to PhD and further on. But we also need a lot of people from the applied universities for these infrastructures.
But the ambition to translate that, we need to educate our youngsters to be the next generation, right? So, okay, fair enough. Others. see what my dream would be if Alex, perhaps?
If I'm allowed to also ask myself or discuss myself. So first of all, there's the most famous German citation on visions is from Helmut Schmidt in his campaign when he became chancellor. Thomas is nodding and he said, wer Vision hat, muss zum Arzt gehen. I think it's not as that anymore in Germany, luckily. I would rather stress, if you're a patient, you need a doctor and a scientist with visions. So I think there we're all aligned. Well, if I just look at parallels, why genomics has grown so much, I think it's not the research discoveries only, but it's really the move into a clinical and diagnostic application. And while I'm visiting the spatial summits with Marcel at the AGBT for the last, I don't know, 6-7 years by now, it's really the strong focus on spatial. There's amazing science and research happening with it, but the real immediate translation to do 10,000s of samples and patient materials in a clinical setting I haven't seen. And I think the market for that is so much bigger. So I think we need to identify the use cases to make a strong move forward, because it's so much easier if your healthcare system, and in particular the Dutch healthcare system, where we have this academic medicine that's almost unique in the world, that allows very fast translation of a latest research tool into a diagnostic and clinical setting in the academic centers. I think that's what we need to identify and push forward, because then you have millions of applications and not only 10,000 amazing scientists that do it.
Cool.
That's my statement.
Cool.
Oh,
yeah, I don't know if it will take 10 years more or less, but I think it would be cool to have stuff that's still alive, because all of these talks start with, oh, we fix it, we take a sample, and to get as close as possible to the original situation would be cool.
Cool. Right, there is seeming a question in the chat. Oh, it's not a question. Great discussion. There's more coming, Sorry. I just need to read. Spatial omics mass spec imaging technologies will be used routinely in diagnostics pathology in 5 to 10 years, complementing immunohistochemistry-based diagnostics.
That's
so
like
maybe
a
little
statement.
bit in line with my statement.
Yeah.
We need to identify the use cases where it can replace current diagnostics, because it's not so difficult. I mean, if it's on par, but a little bit faster or a little bit cheaper with the current diagnostics, we're already doing a great job.
Right.
Yeah, as professor of bioinformatics, I have to add something here. So where really the bottleneck is also the interpretation of that data, and I see still also including in my own studies, the multi-omics studies annotated with KEC pathways, etc., that are, well, very generic, very much lack a lot of content, and so to annotate like these, these metabolic processes in a cell type and tissue dependent context is I think very important, and the same for signal transduction and all the other networks that we're in.
That's like a requirement, right? Yeah.
I think maybe I see also a role here for AI and the large language model that can be translated to molecular context, and I think the first papers are coming out in this aspect. But with the future, I think that will be extremely dynamic.
Yeah. So if I can add my own dream, Fred first, and then I'll take my dream, and then we'll close for lunch.
Sure, we'll keep on dreaming. Ontologies, you should add that to that annotation, because the ontologies are still also not always very good. But my dream would be also to get down to the subcellular level for metabolomics, at least, and lipidomics, because the organellar composition would be, of course, really nice if you could image that, and that is, well, not attained at this moment. So I hope that's a moonshot enough.
Right. Now, and if I may, then my dream to couple different talks and suggestions. Suppose we identify a variant in Alex's long-read sequencing. Well, not Alex's, but Alex's. And we predict, using Danique's expertise, like, hey, this may have a functional equivalent. I would love to have on a spatial resolution, whether it's a cell or a subcell, in a live situation, really live cells, to image what the consequence is. Because we're forgetting cells are incredibly dense. Like, if this stage would be a cell, we would be with 1 million people here. We wouldn't be moving around. No, we're all cramped like this. That's what cell looks like, and how does it interact in vivo and in a living cell. So if we identify a genetic variant, predict what it is, measure it in life, and can perturb it as well, and measure the consequences functionally, and then bring that to the clinic. That will be very cool. That takes 5 to 6 years, I think. And with that happy note, I'd like to thank all of you for the contributions, my colleagues for their contributions, and enjoy lunch, and we reconvene at whatever it was, 1:30. Yeah.